Alright guys, welcome to this video. We are going to talk about how the central nervous system develops. Okay, so the central nervous system develops mainly from the surface ectoderm, right, of the embryo. So specifically, a specialized part of the ectoderm called the neuroectoderm gives rise to the brain and spinal cord. Okay, so after fertilization and early development, the embryo becomes the trilaminar germ disc. So basically here we have the trilaminar, my trilaminar here, germ disc. Okay, so consisting of the ectoderm, the mesoderm, and the endoderm. Okay, so from this mesoderm, a very important structure appears in the midline of the embryo. All right, so this structure is called notochord. So let's look at the notochord. All right, so from the mesoderm, this notochord arise. Okay, and the notochord is a solid rod-shaped structure formed from the axial mesoderm. All right, so it's very important to understand that the notochord is not a nervous tissue and doesn't become the brain or spinal cord. So what it does is that it sends inductive signals. All right, it induces the ectoderm above it. All right, and this ectoderm now thickens to become the neuroectoderm. Okay, so this neuroectoderm now forms a flat structure called the neural plate. Right, so this is the neural plate. Okay, so essentially the neural plate is broad at the cranial end, all right, and at the caudal end is narrow, right? So the cranial end eventually forms the brain, while the caudal end eventually forms the spinal cord. Okay. Now that the neural plate has formed, the next major event is called neurulation. Neurulation. Okay. So neurulation is the process by which the neural plate is converted or is transformed into the neural tube. Okay. And the neural tube will eventually become the brain and the spinal cord. Okay. So what happens is that around day 20 of development, now changes begin uh, in the shape of the neural plate. Okay, so the neural plate, the central portion of the neural plate now sinks inward, forming the neural groove. So as you can see in this structure, there is a shallow depression in the neural plate. Okay, so let's look at another structure to show it clearly. Now look at this structure. We have the neural plate. All right, so we have this depression in the neural plate, and it's called the neural groove. Okay, so these are the changes that occur in the neural plate. So we have is depression in the center portion of the neural plate, and it forms the neural groove. Okay, now at the same time, the lateral edges rise upward. Okay, so these are the lateral edges rise upward, and they are called the neural folds. All right, so these the neural folds together with this. Okay, neural folds. All right, now at the tip of the neural folds, there is a special population of cells that appears, and this is called the neural crest cells. Okay, so at the tip of the neural folds around this side, we have specialized cells that appear, and these are called the neural crest cells. So these cells will later detach and migrate forming most of the peripheral nervous system and many other structures. Okay, so around day 22, the neural folds, okay, begin to move toward each other. Around day 22, the neural folds begin to move towards each other. Okay, and eventually they are going to fuse in the midline. Okay, and converting the neural groove into a tube. This is called the neural tube. Okay, all right, so we have here our neural tube, and after the formation of the neural tube gets separated from the surface ectoderm, and this surface ectoderm eventually form other structures like the skin. Okay, so after the neural tube forms, okay, so its end remain temporarily open. So these are the ends. We have the openings at the ends are called the neuropores. So this is the posterior part of the structure. So here we have the posterior neuropore. So this is the anterior part, and so we have the anterior neuropore. Okay. So around the 25, the anterior neuropore closes. Okay. While around the 28, the posterior neuropore closes. All right. So if the neuropores fail to close, all right, so it leads to some defects like anencephaly for the anterior neuropore and spinal bifida for the posterior neuropore. Okay. Now, so far, we've seen how the neural plates, forms and folds to become the neural tube. Okay. Now, let's talk briefly about the neural crest cells. So, these neural crest cells, they migrate extensively. All right. So, they have their derivatives. Okay. So, they move and eventually become some structures or take part in the formation of some structures. Like we have the sensory and autonomic ganglia. Okay, so let's write them down. We have the sensory and autonomic ganglia. So these are the derivatives of the neural crest cells. We have the Schwann cells. We have the Schwann cells. We have the melanocytes. So we also have the adrenal medulla. Adrenal medulla. So we also have the facial bones and cartilage. Facial bones and cartilage. So we also have the pia matter and arachnoid matter. Pia and arachnoid matter. So we also have the conotruncal septum of the heart. The conotruncal septum of the heart. So these are the derivatives of the neural crest cells. Okay. All right. So before we go further, so notice that the neural tube has a central cavity. Okay. So this central cavity does not disappear. Okay. So even though the neuropores closes, the anterior and posterior neuropore eventually closes. So the central cavity remains, right? And it's called the lumen of the neural tube. Okay. So the lumen of the neural tube in the cranial end forms the ventricles of the brain, while the lumen in the caudal end forms the central canal of the spinal cord. So let's talk about the development of the brain. Okay. The cranial part of the neural tube, all right, enlarges, okay, and forms a series of swellings called the brain vesicles, okay, the brain vesicles. So this happens at around the fourth week, where the primary brain vesicles are formed. Okay, so they include the prosencephalon, which is the forebrain, the mesencephalon, which is the midbrain, and the rhombencephalon, which is the hindbrain. Okay, so and around the fifth week, okay, there is further subdivision, all right, of these primary brain vesicles. So the prosencephalon uh, divides into telencephalon and diencephalon. Okay, the rhombencephalon divides into the metencephalon and myelencephalon, while the mesencephalon, all right, remains undivided. All right, the mesencephalon. Okay, so this is the midbrain; it remains undivided. Okay, so this gives us the five secondary brain vesicles. Okay, now let's look at the adult derivatives of the brain vesicles. All right. So each secondary vesicle develop into specific, adult brain structures. So the telencephalon eventually becomes cerebral hemispheres. The diencephalon becomes the thalamus, the hypothalamus, and the epithalamus. The mesencephalon remains as the midbrain. Okay. The metencephalon becomes the pons and the cerebellum, while the myelencephalon becomes the medulla oblongata. Okay. So this is basically how the neural tube, the cranial part of the neural tube eventually forms, these brain structures. Okay, now let's look at the spinal cord. All right, so the caudal part the of the neural tube, right, is the structure that becomes the spinal cord. So the wall of the neural tube at this region divides into three layers. Okay, so you can see in this structure, the innermost layer is the ependymal layer. Okay, the innermost layer, the ependymal layer. Then we have the middle layer, is the mantle layer, and the this layer forms the gray matter. Okay, the mantle layer, which is the middle layer, forms the gray matter. And we have the outermost layer, which is the marginal layer, and it forms the white matter. Okay, so in the spinal cord, gray matter is central, while white matter is peripheral. So this is the gray matter, and it is central. All right, so this is the white matter, and it is peripheral. Peripheral. Okay, all right. So we now have a groove that now appears inside the developing spinal cord, and this groove is called the sulcus limitans. All right, so this groove is called the sulcus limitans. So it divides the neural tube into the alar plate and the basal plate. All right, so the alar plate is sensory in function, while the basal plate is motor in function. Okay, so this basic arrangement explains, adult anatomy. While why the dorsal horn is sensory and why the ventral horn is motor. Okay, so you know already that the dorsal horn is sensory and the, the ventral horn is motor. All right, so this is because of the the sulcus limitans dividing the neural tube into two functional parts, the alar plate and the motor, sorry, the alar plate and the basal plate. All right, so this is basically how the brain and the spinal cord is formed. All right, so I've come to the end of this video. I believe gained some value. So thank you so much for watching this video and see you in my next video.